My name is Sam Semino, and welcome everyone to the Emerging Technologies Information Sessions 2025 webinar series, week 2. I work for the USGS and the Pacific Northwest Aquatic Monitoring Partnership. And over the last year or so, a planning committee has been putting together this ETIS webinar series, but we did not start from scratch. The Pacific Northwest Aquatic Monitoring Partnership and the Pacific States Marine Fisheries Commission's StreamNet program have been hosting iterations of these emerging technologies conferences and webinars for over 14 years now. So the aim of this project is to bring together monitoring professionals, project managers, and data managers to showcase how they implement the latest technologies in aquatic monitoring. And this year's webinar series will continue over the next 3 weeks with these 2-hour sessions every Wednesday from 9 a.m. to 11 a.m. Pacific time. And I know a lot of you are joining from not Pacific time, so just adjust for wherever you are. And today we have the second session in this series, Artificial Intelligence and Machine Learning. And these sessions are all being recorded. So if you missed last week's session, Modeling and Analysis, you can check out that recording on the PNAMP YouTube channel. You can just go to YouTube, search for PNAMP, P-N-A-M-P, and you probably also need to search for Modeling and Analysis since we have so many videos now. And these videos will typically be available within the week of each session. So hopefully after today, we'll have another video up on artificial intelligence and machine learning. And now I'm going to kick it over to your moderator today, Mari Williams.
Good morning. My name is Mari Williams. I work for Pacific States Marine Fisheries Commission on the StreamNet project. Thanks for joining us for this second session of the ETIS webinar series. Before we get started, I'd like to go through a couple of housekeeping logistics. Sam, do you have the, yep. Okay, on your screen, you should see some of the commonly used icons in Teams. Unless you're speaking, please turn off your camera and microphone. If you have questions or feedback for the speakers, please use the chat function and I can read it for you. Or if you can raise your hand and then unmute when you've been recognized. We can mute you, we cannot unmute you. We are recording the meeting. It will be available on the YouTube channel for PNAMP and a couple other additional troubleshooting tips. If you're having trouble with functionality, try leaving and joining again. Use a different browser. Microsoft Edge generally works better if you're using the browser version of Teams than Chrome does, or if you can use the desktop version instead of the browser version. Next slide, Sam. PNAMP and StreamNet commit to fostering an open and welcome environment free from harassment and discrimination, so please remain respectful. Next slide. So we're going to begin this. Brice is going to talk about artificial intelligence. I'm going to hand the slide deck over to Brice. But as a note, after each presentation, we'll have about 10 minutes allotted for questions, depending on time. And if we have extra time at the end of all of the presentations, we'll open the floor back up for questions on any of the presentations. So as Brice takes control, I have the honor of introducing him. His name is Brice Semmens. He is with the Scripps Institute of Oceanography at UC San Diego. His presentation is going to be on AI for recapture data. He is a fisheries ecologist and conservation biologist. Beyond the realm of fisheries, his expertise in modeling and statistics has led to many exciting collaborations on a diverse set of subjects, spanning monarch butterflies to North American bird populations. These collaborations and associated publications thematically fit into a broader area of expertise, modeling data and innovative analytic methods in order to improve resource assessment and management. Brice participates in a diverse set of fisheries programs, including field-based research fisheries, oceanography along the California coast, and fisheries modeling and stock assessment methods and techniques. He serves in a leadership role with 3 programs that together encapsulate the breadth of his academic focus: California Cooperative Oceanic Fisheries Investigations, CalCOFI, the Grouper Moon Project, and Center for the Advancement of Population Assessment Methodologies, CAPAM. Each of these programs is internationally recognized for excellence in applied scientific inquiry. With that, you have the floor, Brice. Thank you so much.
Okay, great. Hi, everybody, and thanks for the invitation to come and chat with you today. Apologies for the look. I'm trying to use my buff to cover my AirPod Pros to reduce wind noise, so hopefully it's working. I am down here in the Cayman Islands right now, matter of fact, and what I'm going to talk about briefly today is the project that we're working on down here with the Cayman Islands government called Grouper Moon, and it's a long-term fisheries monitoring program focused on this creature right here. This is the Nassau grouper. Nassau grouper is a critically endangered reef fish, listed just so under the IUCN Red List criteria. If you look at catch records from the turn of the last century, it was the single largest finfish fishery in the Caribbean basin. However, over the last 100+ years, the fishery for the species across the Caribbean basin has been serially depleted. And the reason is because this species somewhat uniquely forms massive spawning aggregations, which is what you're looking at right here. And they're very predictable. They happen during the winter full moons, just after either January or February. So January's full moon was just 2 days ago. And while they do not reproduce at all throughout the entire year, they will get together during these full moons and ramp up their sex hormones and reproduce only during these winter full moons, and typically only during one of them. So over the course of an entire year, the entire reproductive output for the species might be over 2 days of spawning at the spawning aggregation. So individuals from many tens to hundreds of kilometers around will, while normally solitary and territorial, will come to specific locations on reef promontories in order to reproduce. Because it's so like clockwork in time and in space, same time every year according to the lunar calendar, and the same location every year on the reef. Once these aggregations are found, they're very easy to fish. Even with very rudimentary fishing technology, just hook and line, you can really do damage to a population very, very quickly, and that's what's happened throughout the entire Caribbean basin. And there are very few aggregations like this left of the species anywhere in the Caribbean. This one is, as far as we are aware, the largest remaining aggregation for the species anywhere by a lot, actually. We estimate now that population is just north of maybe 10,000 individuals. And these are relatively large predatory fish. So a big fish is going to be 50, 60 pounds and closing in on a meter in size. And the island that I'm sitting on, Little Cayman, which is where this aggregation exists on the very western tip of this island, is only about 11 kilometers long by about a kilometer and a half wide. So it's a tiny desert island in the middle of the Caribbean basin. So a remarkable amount of predatory biomass, and it makes you realize what the rest of the Caribbean used to have and no longer does have. So we've been helping the Cayman Islands government to develop and design science and monitoring to help them do a better job of adaptively managing this fishery, because previous to 2001, all known aggregations for this species in the Caymans had been fished out. And it was in 2001 this aggregation on the west end of Little Cayman was discovered. Some say rediscovered. It was a deep past it was known about by the fishing community. Nonetheless, at the time of discovery, there was maybe 7,000 fish. Fast forward 2 years, and just with a few pangas and hook and line, they'd removed the vast majority of those fish from the aggregation. Still, there was over 1,000 fish left, and the Cayman Islands government had seen this story before, so they acted to protect, by 2003, the spawning site, and in fact, all historic spawning sites in the Cayman Islands, and began the Grouper Moon Project to generate information necessary to do better management for the species to rebuild it. And they've been wildly successful at doing so. They've grown this population through adaptive management to perhaps 10 times the size it was back in 2003. Now, looking at that picture and watching that video, you probably get a sense of the challenges in doing population estimation. It's difficult for a diver to get in the water and estimate how many fish they're looking at. It was easy somewhat early on in the process when there was only 1,000 odd fish, but when you get that many fish, you can never truly see the extent of the aggregation. It's deep diving. It can be cold, it can be rough. So we need to do something better than just, hey, divers, estimate how many fish you've got. And for a long time, we've been doing mark recapture using external FLOI tags, which is what you're looking at here. And here the idea is you tag a small portion of your fish in a population, then you go down with divers and you count numbers of fish, let's say 50 fish. And of those 50 fish, you count over and over again, randomly, as best you can. You estimate which of those fish have tags, not estimate, you count. So, and you do that over and over again. And through those data that you collect over and over again, many counts of 50 fish, and of those, how many have tags, you can get a proportion of the counts of fish that have tags. So if 5 out of 50 fish are tagged, then 10% of the population is tagged. And if we've tagged, let's say 100 fish, and we know that 10% of the population is tagged, we can estimate or infer that the population is 1,000 individuals strong. So that's the basis behind the Lincoln-Peterson estimator, right? This works great. It's been effective for us, but there's challenges with it. Not the least of which is we're sticking jewelry in the backs of critically endangered reef fish. And as careful as we are, there is no question that this has some impact on individuals. Sometimes things go wrong, infections happen. So there is a cost to doing it. There's also a cost to the local tourism because people like to see pristine reefs and they don't want to look at a top predator with a pink tag hanging off its back. So we want to get rid of that. But most importantly, what we can tell from these tags, they are numbered, but you can't as a diver see the numbers on these tags. So the best we get is the animal tagged or not? Well, that doesn't give us individual information that allows us to identify that individual in a repeated way. And because of that, we can't easily get at things like growth rates and individual mortality or skip spawning. What proportion of the population aren't going to spawn each year? Are individuals associated with each other on a regular basis? All of these questions that we can get to if we have individual data, we can't get with this tagging data. And so what we've transitioned to over time is this notion of facial recognition. So it turns out, like you and I, Grouper can take on characteristics, visual looks, in this case patterns, that are consistent and persistent. So they last from 1 year to the next, from 1 month to the next, 1 day to the next. And so we can use this pattern and facial recognition or pattern recognition tools, individual instance pattern recognition tools, to keep track of individuals based only on video data that we have or images that we generated from these fish at the aggregation site. So this, by the way, is what ChatGPT thinks fish facial recognition looks like. So there you go. ChatGPT for the win. That's not an NASA grouper, by the way. It looks more like a Goliath to me. And I don't know what Instagram has to do with that, but ChatGPT thinks there's something. How are we doing individual instance pattern recognition? Well, we partnered with a nonprofit organization by the name of WildMe, which has subsequently been subsumed into Conservation X Labs. And they are an AI and tech nonprofit that brings to bear computational techniques and AI tools to inform conservation and management. And they have this program called Wildbook, where for a bunch of different species, from zebras to cheetahs to, in our case, groupers, they've generated a back-end workflow to do individual instance pattern recognition based on libraries of images to allow researchers to do exactly what we're hoping to do, which is track individual fish across time in a manner that allows us to leverage mark recapture tools for population level estimation, as well as things like growth and survival estimation. And I'll say it works. So here's an example of a individual that was captured on video about a month apart. And if you kind of stare at it long enough, you might start to see some of the ways in which this creature matches. So here, what looks to me like a map of North America exists right here. There is this sort of constellation of, it looks like the Southern Cross. You get that here. So once you kind of spend some time looking at it, it's very clearly the same fish. And here are the areas of high match according to the individual instance pattern recognition tools that WildMe has. And here's a creature that is 7 years apart. So the image on the top was captured in 2012, and the image on the bottom was captured in 2019. And again, if you stare long enough at the patterns on the side of this fish, you will notice that it's fairly obvious that this is a matching individual. But here's where AI tells us that it thinks it's a match. And so upwards of 7 years apart, and probably a lot more now. This is just with some preliminary data that we started the program with. We're able to capture the same individual across time. By the way, these creatures, Nassau grouper, they can live to 30, perhaps even 40 years. We have documented through otolith analysis through the Cayman Islands, 31 years is the oldest individual. So they'll live a long time. They get reproductively active, or they start going to spawning aggregation somewhere between 5 and 7 years of age. And I will also point out on this earlier one, you can see these little red dots there. That's actually laser calipers, which we can use to do size estimation, and we'll talk a little bit more about that. So here's the front end of the platform. We've not only used this for our work at the spawning aggregations where we dump a whole bunch of science images, but we've created an online portal that allows citizen scientists such as yourselves, should you so desire, to take a camera into the water, snap a picture of a grouper, and upload it to a portal where we can make use of that information to keep track of individuals not during spawning season, for instance. And that's called GrouperSpotter.org. One of our challenges is been recovering all of the images from our old videos and imagery. We've been doing this since 2003. And so we've got 21 years now of images. And just to show you on the left, that digital video tape, that was the hot tech when we started this thing. It was brand new to the market, consumer grade DV tape video. And so that camera below, that's our laser caliber setup with the DV camera in the middle of it. We had these 60-minute tapes. And of course, we've come a long way since that. GoPro blows this stuff out of the water at this point, has for a long time now. But we have many tens of terabytes, thousands of hours of images and we a video, and we need to go through that and weed out individual images of fish in order to populate our database, our library of images. And right, and I showed you the caliper video, where nowadays in a modern context we do stereo video, and size matters. Of course, in a fisheries management standpoint, the two key pieces of information is how many fish and how big are they. And we're using stereo video, but we've only been able to generate length frequency data. We haven't been able to track growth of individuals, but with the potential for tracking individuals across time, plus the stereo video data we now have the ability to capture an image of Bob across 5 years, and Bob the fish might have grown 5 centimeters in 5 years. So we have that as a potential for useful information for understanding how the fishery is working. So what we're doing with the individual image, pulling the fish out of the videos, is we've trained a YOLO V9 fish detector with a team of scientists and engineers on upper campus at UC San Diego, which is where I'm at Scripps Oceanography. And we're working with them to train boxing algorithms to do fish detection, and we've actually improved this quite a bit since then. But even with our sort of first round of training and library prep, we're getting pretty good detection rates for boxing of individual fish. And the plan with our video is to segment those videos into roughly every half a second, and then do boxing on each frame of that video across all of our data. The challenge, though, is that especially for the more modern videos that we've got, this is what the images look like. And so you've got a challenge where the AI can potentially get swapped. The YOLO identification tools apparently are not working especially great at capturing all of the individual instances of fish in this image. And so we're working on methods for trying to get the detector to do better when there is a huge number of individuals in a frame. And so this is from one of the more advanced or modern cameras that we've got in the water. And I just wanted to point out, first of all, there's a lot of fish in this picture. If you just zoom into this section right here, only that section of this entire photo, and you zoom in, well, you've probably got upwards of 20 individual fish that have enough resolution of patterns to do individual instance pattern recognition. So we can get hundreds of individuals per frame, and we can get many thousands of frames per half hour. And so we computationally have a very challenging problem ahead of us, and we're trying to tackle that. So this is one of our challenges, is getting folks to do library prep for training the YOLO detector. So when you have a frame like this, we have to have some poor soul sit in front of a computer and do boxing for hours on it. But fortunately, we're starting to make progress on this. It's not easy. And to that point, I found it challenging to find generic fish underwater training data sets. I know there are some. I know that NOAA has one. I know Viame has one, but none of them, near as I can tell, or very few of them, are publicly available in a nicely organized and curated way, which is unfortunate and something that we as a community need to work on. The other problem with those many thousands of frames of an individual is we don't want to swamp our individual instance pattern recognition software with 500 images of the same fish that all look like this, right? So looking at this fish, we've got maybe 70 frames of the same fish. They all look the same. We can't send all of those to individual instance pattern recognition. We just computationally, it's impossible for the algorithm to deal with that many individual frames. So our next step is to try and figure out tracking of individuals, and then ultimately decide algorithmically how to choose which frames to use, subset, maybe 3 to 5, but not 50, right? So how do we choose those frames such that they're a good representation of individual, but maximally different from one another to have the best information set forward to inform the model. So we've been experimenting with Viame, and probably some of you are familiar with this. This was I'm going to get the details of it wrong, but it was originally a NOAA inspired or NOAA built tool, which is now being, I think, curated and maintained by a company. It is somewhat open source. I say somewhat because in our experience, it's been a little bit of a challenge to look at the documentation. It does fairly well right out of the box at boxing fish, and you can train the detector to do boxing. It does have a tracker system in it, so you can track individuals, but you cannot train the tracker. So you get what you get there, and you don't throw a fit. And so we haven't spent too much time testing the tracker yet, but we've kind of left that for now, just because we know that we can't do any training to make it any better. With Viame, it is what it is. That's just what I said. Okay, and then this question of selecting a few frames from the hundreds of individuals, what we've gone down the road of is first training a YOLO to do boxing, and then using segmentation tools, AI segmentation tools. There are two that we've tried. Fishiol, which is a built for purpose fish segmentation tool, and the other one is Google's Segment Anything. And we're still in the process of testing out these two segmentations. If you just use segmentation on a whole image, let's say that image of those many hundreds of fish, it does okay for the fish in the foreground, but the background, it kind of blows out. So what we've decided to do as a workflow is to do boxing first and then segmentation on those boxed and cut out individuals. But that's a work in progress. So stay tuned on that. You can train both Fishio and they're both open source, I believe, and segment anything. Yeah, so I'm not sure exactly where we are on the training step of that segmentation work. But the point of this is, once we get segmentation, we can determine algorithmically, we can determine orientation of individual, and then we can decide to choose individual frames from the wealth of frames that we might have for a single individual in order to have the best chance of doing individual pattern cross-matching and also providing as disparate as possible types of looks of the same individual to help for future matching. That's the goal. So that's I think what eventually is decide on those morphometric metrics that allow us to do that frame selection. And then finally, I'll say that we're expanding GrouperSpotter to include not just Nassau Grouper, but also Goliath Grouper. So we're in the process of building out training libraries to train up Wildbook tools for Goliath Grouper. And we're also over in the Pacific Islands, we're doing Camouflage Grouper, which like Nassau Grouper for massive spawning aggregation. So this is footage that was shot by Tom Spark over on Fakarava in French Polynesia. And we have the same problem with Nassau Grouper, but it's kind of the same problem, but on steroids. So, I mean, here's an image of Camouflage Grouper. This is not doctored. This is an actual image of the spawning site of Camouflage Grouper in Fakarava. So, I mean, you can imagine how AI is going to be challenged to pull fish out of an image such as this in an effective way. And so this presents a future challenge and opportunity for us as we move forward in this journey. And then, okay, the other thing I wanted to talk, I know I'm almost out of time, so I wanted to just say, we're also developing underwater tools that are low cost, low impact, but allow us to capture length estimation of fishes. It's through a program called SMILE, or Size Matters Innovative Length Estimation. And it uses a consumer grade point and shoot camera and a single laser. And it turns out that through a single laser, and where that laser falls on the objects in its frame of view relative to the pixel dimensions of an image allows us to estimate distance away, right? So if I've got a laser to the left of my camera and the laser hits an object in the far left of the image, that's because the object that it hits is very close to me. But as the object that the laser hits moves further away, it's because it's closing in on that vanishing point that's very far away from us. So we can use single laser dot in the pixel dimension to actually calculate the distance to the object that we are hitting with our laser, as long as we calibrated the laser and the camera lens together. And so we're using this somewhat simple math in order to use these single laser camera setups to capture length data. So here's an image from our labeling studio here. We're working through some images, and that's a blue parrotfish in the Florida Keys. If you squint, you can see that there's a red dot on the center of it. That's our laser. And so we're using fish field segmentation to segment that fish. We're using a laser dot identifier AI tool, which we're training because it doesn't work great. That's a challenge. It allows us to document the location of that fish. Once we segment it, we can also use AI tools to mask it, identify the fish that's being shot by the laser, and then put dots on the tip of the nose and tip of the tail, all again, algorithmically, automatically, and then ultimately come up with a length of a fish. And so the idea here is to put these in the hands of a citizen science marauding horde on our reefs to capture information for those very data deficient species that we care a lot about. This is a hogfish in the Florida Keys. Hogfish are, of course, a very important and somewhat data deficient species from Florida. So this is a great example of a species that we care a lot about, and divers generating information on them would be a very good thing for fisheries in order to help them to build better models of what's going on with the stock. Okay. I will stop there, and this has been funded by lots of different folks, including NSF, Office of Naval Research, big partnership with UCSD, Engineers for Exploration, and NOAA, and all of our partners down here, most especially the Cayman Islands government. Okay, thanks.
Thank you, Brice. Technically, you went pretty fast. So we have about 15 minutes possibly for questions before we move on to the next presentation. And I want to say that camouflage grouper is well named. That's about.
Yeah.
Great presentation. Thank you so much.
Absolutely.
Minh, do you want to unmute and chat?
Yes, thank you. Your work is really amazing, Brice, and I wonder how long did it took for the AI side to train to be able to detect all of this? Because the work, especially when you were talking about the slides where somebody had to sit there, probably spend the entire co-op term boxing all of the fishes in the image, that probably sounds like a whole term work. So just wondering what was the time scale for that?
Yeah, we're still doing the training, building the training data sets. That has by far and away been the biggest challenge for us is the human part of the work. In particular for those massive images where you get 100 or 1,000 fish in a single still frame, that boxing, it'll take somebody a couple hours per frame, and we've got tens of thousands of them. So that's been a real challenge for us. And what we've done, first of all, I pay a lot of undergrads, and I also have some undergrads that are doing 199 projects, so they kind of make a project out of it. And we partner. All of this is done in partnership with the Reef Environmental Education Foundation, or reef.org. And Reef is a citizen science nonprofit that is one of the co-leads on the Grouper Moon project, and they are also the world's largest profit. Their fingertips, a wealth of not only people that are willing to volunteer their time, but people who know a lot about the fish they're looking at. They know the difference between a Nassau grouper and a Goliath. They know what a blue parrotfish is. They know the difference between a midnight and a blue, and that's hugely valuable to actually have people who not only are able to box, but they know what they're looking at. So that's been a big asset to us. So that's, I would say, to answer the question more succinctly, the time suck for us, and it's been a couple of years, has really been library prep. And that's why I think it's unfortunate that there isn't a more well-organized and curated set of generic training data sets for boxed fish. We got throughout NOAA and the broader and international fisheries management community, there's a whole bunch of people that are using Viame, but you can't find their training data set. Everybody's building their own data sets and training data sets, and nobody's sharing. Like, it's crazy. So, man, we got to do better at that, everybody, collective us. We got to get better.
Well, we've got 47 people online here now, and I see the views on YouTube popping up, so hopefully we'll get some collaboration happening.
Yeah. Somebody needs to be the champion and organize us.
Yes.
That's always the thing, right?
Yeah. Yes. Do we have any more questions for Brice? You can enter them into the chat, or you can unmute at this point, and feel free to raise your hand or whatever. Okay. So Brice, are you able to stay on with us through the rest of the presentations to potentially answer at the end? Your contact information will be available through both the posted video and in PNAMP information on this session.
Yeah, I might have to go at 1:20 to go catch a boat out to see the fish and clip some more video. But up until that, I'll hang around.
Awesome. We really appreciate it, Brice.
Oh, thanks for the invite. I appreciate
Yeah.
it.
Okay, so next we have Tom Bird and Minh Doan as they take control of the presentation. I get the honor again of introducing them. So Minh Doan has been working with salmon data analyst has been working as a salmon data analyst at Canada Department of Fisheries and Oceans with Tom Bird for more than 1 year now. Minh works closely with the salmon data, historical and present text and images, and integrates AI to help bring salmon data to the bigger world. His work varies in methods from image recognition to AI prompt engineering for reports and documents to clustering and text analysis on multiple sources of documents. Tom Bird is a research scientist at Fisheries and Oceans Canada. He likes being able to find things, particularly data things. At DFO, he is working to help DFO science improve how it works with data at all points in the data lifecycle. He has worked in research, consulting, and large NGOs using different types of survey data to explore the distribution and characteristics of human and animal populations. He is currently focused on figuring out how investing in enterprise and cloud development environments. He is particularly interested in rescuing and publishing dark historical data assets, and improving interoperability between salmon data sets. And with that, Minh and Tom, take it away.
Thanks. I think Ming's going to share his screen. I'll do the first part, and I'll do the easy part, and he's going to go on the hot seat for a demo. So thanks, Mari. Nice to see you and other familiar faces here. And we're going to talk about words as data and how to use large language models to go through a lot of words and analyze them. So I started my career in fisheries as a field biologist. Let's go to the next slide. And in the last presentation, we learned about Bob the grouper. In this presentation, we're going to talk about Bob the biologist, which is, as I said, where I started my career in fisheries science. And we're going to talk about a person who is at a governmental organization such as Fisheries and Oceans Canada, or maybe works with PNAMP or the US Geological Survey. And maybe they're responsible for monitoring the health of fish habitat in streams. And we know that there are hundreds or thousands of these people distributed around our respective countries and organizations. And they share at some point in their careers, they each share a common problem of, given the job of monitoring a fish population or a habitat, they wonder to themselves, what method should I use in order to meet my objectives, the job that's been given to me, and given the constraints that I've been given. And so typically what Bob might do, let's go to the next slide, is go to the literature. And they might say, all right, let's search for research papers on monitoring fish populations. Or they might talk to somebody who came before them, and that person might say, well, here's the method I used. Basically, they have to do a lot of reading and assimilation of a whole bunch of different sources of information. And that biologist has to look through all these things, figure out how these things relate to their work, and see whether they can use them to meet their goals. And on their journey, they might read a paper about capture recapture. They might see this cool thing about eDNA. They might read a paper from the 50s about netting off a whole river and electrofishing. all the fish inside that thing and not caring about whether they kill them all or not. And they might not understand all the things that they're reading. Next slide, please. So luckily there are existing repositories of methods. And as I said, like sources of literature online. But again, they represent thousands of methods, for example, in monitoringresources.org. It's a goldmine of knowledge, but it's a huge volume of information to sort through. There's a lot of diversity of information, and a lot of it is highly complex and maybe outside of their experience. But really, all this person is doing is a few key tasks over and over and over again. And it's just unfortunate that they happen to be kind of slow at it because it takes a while for the information to go from the screen or a piece of paper through our eyeballs into our brain, into processing centers, all that stuff. And unfortunately, we're not very fast or consistent at those particular tasks, which in themselves are not hugely demanding. Let's go to the next slide. And so this is where we can use AI and large language models to help because we can represent words in a digital format. AI can read them very quickly. It's not actually thinking, so it's not biased by the content, and it has no opinion on anything that it's reading. It can do this very repeatably and easily, and it's becoming increasingly cheap to get a computer to read your stuff and then analyze it. And that leaves our friends, the biologists, to go and do the things that they love doing and are good at or better at than machines, like jumping into the water to go look at groupers, which I'm very jealous about. Next slide, please. So next we're going to go to the hard stuff, which is Ming's job, and he's going to tell us about how, like a sneak peek at how AI works with text data.
Hi, everyone can hear me?
Yes, we can.
Okay. Thank you, Tom, for that. And I'd like to say that, because AI is cheaper and able to read things unbiasedly and objectively, how does it do that? Well, the sneak peek is that AI turn all these texts, these words that we'll look at, into numbers. So, for example, the word fish, for example, can be converted into this mathematical vector. Here's an interesting tidbit: is that this is about a third of the length of the vector. I can get that all in, and then at that point, we'll probably just see some squiggly line from top to bottom at that point. And then the longer the vector is, actually, it enables the word to become more and more precise and distinctive, so that it can then use that to analyze all of the text data together without being biased. And then based on these tokens, because in math they're vectors, so it can do many things with that, because we have explored all that in mathematical formula and matrices, linear algebra. And then, so two things that we're taking advantage of AI be able to do that is summarizations, where at the core of it is finding the average between all the vectors, and then generate, and then that's where the generation comes in, where it can then generate a summarization that encompasses all of those vectors, or clustering, where it can apply the graph theory and then able to find the neighboring vectors together, and from there on form each of the clusters. And because this is all data driven in that sense, it would have less of a bias from the main experts' structures that you usually studied off if you're coming into it from the scientific viewpoint or from the academy viewpoint. So what AI will do for Bob is that they will do all the reading, so Bob doesn't have to do the reading, and instead of going through all the data, and Bob doesn't have to, AI will go through all of them for him, and then AI will also do all the summarization, so that will shorten the amount of information, and it will also convert all of the informations from differing level of details and expertise that is required into the same level. Also, and then for clustering, AI can do all the tokenization and then find also those clusters. And then all Bob needs to do is just look at the list of available topics. Oh, are they separate enough? Are they independent enough? And then Bob can go into each of those individual topics to find what he's interested in. That greatly simplifies his time so that he doesn't have to read a lot. He just needs to read a few. And of the things that he reads, he doesn't have to read them at their raw level of different level of difficulties. He can just read them all at the same level, making it much easier to compare between different methods that he finds. And so here's where I'd like to present MAS. This is basically our interactive dashboards that we're currently built. You could say that this is maybe our pilot of what it is. It's still going under many changes, but this is where we have gone to. So based on monitoring resources, its own data has thousands of methods available. We've been able to categorize all of the methods into about roughly 30 different topics. And interestingly, yes, there's also transponder pit tag in there too. And then this is the graph of the transponder pit tag. Of this one topic, there are all these green that are noted as methods. And then from each of them have these protocols, a group of methods that are used. So in this way, if Bob were to search for transponder pit tag, he'll be provided with all of these lists of methods. And then from there on, he can find that, oh, maybe I work in a large river. Then he can just select this method. And then he can go in, and then he can read that in details. Each of these topics also has a list of words that are kind of like keywords of this topic. They represents the most information. That represents the most standout information in this topic. Allowing Bob to, he can go through different topics and then finding out which one best corresponds to what he does. And so with methods, for example, here what we have is that we have lists of most commonly used method. So even if Bob doesn't have an idea of the topic, for example, maybe what he needs is very niche from this list of available topics, he can go into these methods here and then go through them to find. Currently in this one, the commonly has about 50 of them. It's not limited to any of there's not 50 in there, it's 1,500-ish. And then Bob can just find in here. So for example, we were looking for counting salmon reds in shallow water and large river. So we can find that methods, and that methods, we have a number here showing that. But that's the less important part. Here's the more important part, because we're able to summarize the whole method into one abstract. We're also able to, from that context, able to see if there's any constraints. So for example, this one just described here, and then there are about 3 constraints in here. One is the water depth constraint, there's a red appearance constraint, and depends on the watershed size. So he can just glimpse through all of this and find that. But what happens if Bob already read through all of this and be like, okay, I'm interested in that, but what exactly does the method read? These summarizations are great and helpful, but if he can't read the raw thing, then he can't use it. So we have the description part here that has the details of specifically how it's done. And then each method, we also bringing it in with more keywords and tags that are auto-generated by AI, so that it doesn't have to rely on the person who initially wrote this to put in their keywords. Because somebody's understanding of salmon can be different from somebody else's understanding of salmon, sadly. And because of that reason, sometimes if you're searching for one specific ideas, it might not correspond to another person's, and therefore missing out these things. Giving this job to AI to be able to identify out these keywords would allow them to have a much more common base with every method in the entire database. We have the original ID to go back if they want to, and they can have the short written target of these methods. And last but not least, if you created all these keywords, these tags, well, what can you do with them? You can provide them a list of tags here to assist Bob in searching this information. So if you're interested in something, so let's say water quality, he can immediately find methods that containing the water quality, and then from there on, he can search through there, assuming that he can't find from a method or he can't find from a topic, he can find from the word itself. And that is currently our work in building this method support system, which is called MAS. And we're using this to help biologists to be able to go through databases of text data. And this this relies entirely on text data. So if you're not working as biologists reading biological paper, you can also use this to read, let's say, in health or in cybersecurity, or even in police related work, where the main bulk of the work has been going through a lot of paper written reports and then be able to find out what you need from them. And the problem is further exacerbated because of the different people writing it, especially because you have different people of different level of expertise. That brings in the need for a more standardized approach to be able to find what you need. But however, as said, this is still a pilot, and we're able to find that there are struggles that we have to overcome. So one of the things is that our summarization, while come out is easy to understand like that, it could potentially be missing important informations, or it could entirely be mis-summarization because of those ideas. So that's why sometimes you get a subject matter experts to be able to verify some of them, or ideally is verify all of them. But our idea is that we could find perhaps be able to verify a much smaller amount and then from there project onto the rest, or have another AI to verify that. But then that AI has to be verified by a subject matter expert. It's kind of a problem between the chicken and the egg. And then another thing that we're also developing right now is our searching capability. As said there, one more problem that Bob could be facing is that Bob could be entirely a new biologist. He just came in from Environment Canada, for example. He's been working with trees and forests the entire time, and now he has to go to fish. While they're biological, it's not the same thing. So Bob has to be able to adapt what he knows in order to work in here. So semantic searching is an idea where, similar to Google search, you type in what you want to search, and then from there on, they try to find you the most similar things to be able to provide for you. The idea that we have these topics in our database during that presentation was great. For an idea, if you need to find something related to that topic, you can always find it within that topic list. But if it's not in there, or if you need to find something more specific, semantic search is the better way of searching. And last but not least, the searching would also need to be able to be personalized and stored for each user, so that when they search for something, they can keep the result like that, so that they can find it later. And that'll be all. Thank you for listening. That has been Mass from the Department of Fishery and Ocean from me and Tom. But we'd like to acknowledge that we had our help from our cloud from Shared Service Canada and the people from Monitoring Resources, such as Sam and Jen. Unfortunately, I don't see Jen here. But yeah, thank you very much.
Thank you so much, Minh and Tom. That was a great presentation, and I really love the keyword generation, because that is always a block point for me. So we'll open up the floor if anyone would like to unmute and ask questions of either of these gentlemen. We welcome interaction. I don't have anything in the chat right now.
Yeah, I have a quick question. So what do you guys see as the next steps for mass or for large language models in general when looking at these texts of biological data? What do you want to see come next, essentially? Or what are you planning on doing next?
I could take a stab at conceptually what we're looking at doing, and Minh might want to jump in as well. But one of the most important things to helping tie together all these different types of information is some sort of formalized description of things, like the ontologies that describe what is this thing and how does it relate to this other thing. So that is something that we're actively working on with colleagues in the Salmon Data Mobilization Working Group, and we're looking forward to having a workshop on that in March, which we're all invited to, I'm sure, at the American Fisheries Society conference. Shameless plug there. I'm not sure if pets are invited, but we can ask the organizers. And shoot, what was the second thing? I forgot what the second thing was, but I'll let Minh jump in on what he sees as sort of next technical step.
Thank you. I think the second item talk about that is basically how do we also train the AI on pre-existing data. I think some methods are very well documented, and they can be noted as, this is the standard that we expect from a golden method, for example. And then from there on, all the other methods or other documentations can also be written in that form, such that it's more concrete across the board. Certain methods noted are also a little bit I wouldn't say poorly written, but they lack sufficient information to be able to present to everyone. So having an AI, after it's being trained on other methods, especially other similar methods, be able to replicate and then add on those information, that's that would probably be a next step on that. And then not just the training part, but also we'll be able to adapt to more databases at once, so that when it goes through databases and find the most appropriate methods for a biologist here, that would be the most appropriate, probably, expansion of this. It's not just one system anymore, it's going to be multiple systems. So that's our that's our development plan, I believe.
Thanks, Minh and Tom. So potentially, this AI machine learning could, in the long run, help users document actually their own methods by it reads these golden methods that have already been created, and then it could potentially create a template for those that come along next to help them identify what are the key pieces of information that would be needed to, then group this method with other methods or compare methods to each other. That's approximately right?
Yeah, that's correct.
Very cool.
Okay, folks. Do we have any more questions? All right, so we will move on. Minh and Tom, thank you so much for the great presentation and the plug for the AFS ontology workshop at the WABC meeting in Vancouver in March. We're going to move on to our next presenter, Aaron Legge. And while he takes over the slide operations, I have the honor of introducing him, and he is with Innovacy, and he is the head of excuse me, the head of project management fish tracking at Innovacy. Innovacy is a pioneer in developing end-to-end solutions for aquatic ecosystems in the most challenging of conditions. Aaron is based out of Halifax, Canada, and has been with Innovacy for 6 years as a product manager. The floor is yours, Aaron. Thank you so much for being here today.
much for having me. Absolutely, yes. Excited to be here to share information with this group on a new product that we've been developing here at Innovacy over the last number of years. Really focused on improving hydroelectric dam operations and helping with the monitoring and the counting of fish moving through hydropower. So a bit of a background. Who exactly is Innovacy? So Innovacy, our head office is in Boston, Massachusetts. As mentioned, I'm part of the fish tracking business unit. The fish tracking group is based in Halifax, Nova Scotia, and we've been around for quite a while, 30 plus years of history with monitoring and tracking fish, and might be better known to some of you who have used acoustic telemetry before as Zemco, the former name of the fish tracking business, now part of Innovacy. So we've taken that experience that we've had with fish tracking and acoustic telemetry and the work that we've done around a number of different hydro facilities as part of that product development. And we've launched a new product here that we're calling Hydro AI. So Hydro AI is leveraging high resolution video cameras and cloud-based algorithms to do accurate real-time fish counting and species identification as fish are moving through fish ladders and other infrastructure as part of hydro dam facilities. So in our research for this and kind of getting started, we were talking to lots of hydropower operators and certainly realized that there were a number of challenges that they were having in maintaining compliance with their regulatory operations. And they were going through manual fish counting processes. I'm sure there's some folks here who've had experience doing fish counting and kind of having to manually count, watching fish or taking sampling. There was lots of data that was being collected, but it wasn't always able to be processed. Generating reports from that count data often took a lot of time as well and could be rather tedious. And in situations where that reporting was really taking a long time and not being as accurate or as precise as it could be, some of those hydropower operators were seeing times when they may have to shut down or they have to curtail green power generation because of the uncertainty around the data that they're collecting of how they're impacting those migratory species moving through hydropower facilities. So our system contains a number of different components. So we've integrated the camera system into some fish ladders. So you can see kind of in image number 1 there on the left on this slide. I'll go into a little bit more detail about the specific sites that's installed in, but the camera is kind of installed in that structure. It's looking down into the fish ladder, as you can see in image number 2 there, and counting and identifying fish as they're moving through the fish ladder. Image number 3 kind of shows the on-site component of our computing. So there's a box that we install on site near the camera that contains all of the internet connectivity. It's supplying power to the camera. It's really kind of the brains of the system, moving that data around. And then, as you can see, lastly, in number 4, we're then reporting all of this data in real time with a cloud-based reporting software platform that allows users to look at a number of different ways of looking at the data and accessing that remotely in real time. So all of those components are kind of running together. As you can see, the camera is obviously counting 24/7, which is an exciting kind of advancement we can make by having a camera that's sitting there and doing it continuously. There's no need to sample and kind of extrapolate those numbers from samples, and no need to just kind of only be taking at certain times. We're able to look at an entire data set for when the camera is installed. We have an edge device that can do some processing as well as kind of pushing things to the cloud. And then we've also been working with our partners in incorporating acoustic telemetry studies in order to validate some of the insights that we're seeing as part of the data that we're processing with Hydro AI, and also exploring some ways to leverage aerial sonar data to kind of understand those larger scale movements as opposed to looking at the very kind of fine scale data of individual fish counting moving through the system. In terms of the software, I've got a few other screenshots a little later on as we move through, but as mentioned, that's happening in real time and coming from multiple cameras. So in many of the sites that we've been installing here in Nova Scotia, we have cameras at the top and the bottom of each of the ladders. So we're able to count kind of animals moving in both upstream and downstream directions at both the top and the bottom of the ladder to try and get an understanding of the actual number of animals that have moved through. So this site updates every minute with the new count of fish that have come in the last minute from the various cameras. There's an ability to go back and look at an archive of video. You can drill down into a particular day, a particular hour from within a day, right down to a particular minute of the day, and watch the 1 minute video clip of that camera recording to see the fish that were identified by the model, as well as exporting reports directly from this site of those counts and being able to share those internally, or obviously share them with regulatory partners as well. This particular camera housing was designed for working with our partner Nova Scotia Power here in Nova Scotia, Canada. They have a number of these pool and weir style ladders for river herring that are migrating through dams in their power system. So this is the structure that we designed for that. As you can see, there's a bit of a metal base here that allows for water flow to kind of smooth out a little bit, make it a little easier for the camera to get that view. The camera is mounted in the very middle of the top of the housing here. You can see the wire running to the camera, and that just gives us a consistent field of view, a consistent background, consistent lighting, all of those things that make developing a model for identifying things in those videos a little easier by having consistent video as we're moving through. So as mentioned, we've been working quite a bit with Nova Scotia Power as we've been developing this, not only because they're right here in our backyard, which makes things a whole lot easier, but they were also a great partner with us as part of an ocean supercluster project. So the development of Hydro AI started as a project under the Canada's Ocean Supercluster, which was a funding program for a number of different innovative programs in ocean sciences. This was a project that we were working on with Nova Scotia Power called the Ocean Aware project. Started in 2022 was our first installation at Nova Scotia Power. And we deployed the system as well as a significant tagging study of these alewife, or Gaspereaux as they're known in Nova Scotia. And when we compared those to the acoustic telemetry kind of movement numbers, as well as comparing it to the numbers that DFO was collecting as part of their own counting program around White Rock, we were seeing a greater than 95% accuracy in the numbers that we were counting, and we expanded that partnership with Nova Scotia Power this year to 8 additional sites across the province. So a little bit more detail here on that first installation. So at White Rock with Nova Scotia Power, it's on the Gaspereaux River here in Nova Scotia, built in 1919, powered up in 1920, and does contain some fish ladders and downstream bypasses to allow those alewife to move through. Rather significant population of alewife in the area, so somewhere between 500,000 to 1 million fish every year migrating, and there is a robust both commercial and recreational fishery for the alewife. So here's a kind of an aerial shot of what the White Rock system looks like. So the river is kind of flowing. I'm not sure if you can see my mouse, the view that I'm in, but kind of on the, you can, perfect. Yep. So river's kind of here, fish are swimming upstream, so they're coming up the ladder. And what we're seeing, the generating station is way down this canal. And obviously we want to see the fish continuing their migration this way. So what we installed as part of all of this system was there was a camera at the top of this ladder. There's a camera in the fish bypass, and we had 2 ARIS cameras installed on either sides of the outcome of the fish ladder to get a sense of movement, of directionality as they're moving in that canal. Are they traveling in the direction we want to see? So that was the setup. This is a bit of a view of what the camera inside that fish ladder sees. So as you can see, this is looking down directly. This metal plate under here was the one that we were seeing the side view of in the overhead view earlier, and just the kind of as the fish are moving through, the algorithms counting them. This particular application for Nova Scotia Power is in the order of kind of 99% alewife or herring moving through here. So we were not doing individual species identification as part of this project. This was just, they were trying to get an overall number of the fish that are moving through the system. We've started to do some algorithm development as we've expanded to some other partners that we're working with and collecting some more diverse data sets. We've also started working on some new models for doing things like speciation, as well as just identifying anomalies in some of those data. So we were able to look at the Nova Scotia Power data set, and we identified a number of fish that were larger or smaller than we expected alewife to be as part of the processing of the video, and we're able to highlight those and flag those both for Nova Scotia Power and their regulatory partners to manually review that video to determine what those species might be. As we move towards trying to do a lot of that programmatically with our own algorithms, I've just got to, it's not going to play because it's an MP4. That might be the case. But as you can see here, what we're working on right now is a trust network based on key point analysis. So these are some salmon parr from a site in New Brunswick. And we are starting to develop a model that can identify these key points, build these trust networks, and then we can use those for doing speciation of the fish as they are also being counted and moving through the system. Obviously, this particular screenshot here is not from a video of a fish within a fish ladder, and it's really to make this key point analysis and this trust network model successful, we need this side view as opposed to the top-down view. So we're working on new ways to install and cameras that we can install to get these side view. We know that there's lots of sites already doing some video counting or at least capturing of fish passage in an attempt to do that counting with a side view. So this is really kind of where we're moving in terms of our model development. But once we've captured all of that video and the algorithms have done their thing, when we're spitting out the data, because we're capturing 24/7 data, we have some really interesting things that we can look at from that data that hasn't been able to be really kind of looked at before when taking statistical sampling and kind of extrapolating those numbers. So we can go from an hourly count and really kind of looking at where are the peaks and valleys. You can see here, this is kind of the daily cycle over the course of a week of the number of fish that we were counting at one of the sites with the two cameras, camera one being bottom of the ladder, camera two being the top. And we were seeing a very interesting pattern that we've seen over the last couple of years with these alewife of having what we jokingly call kind of morning and evening rush hour traffic of their own. There always seems to be a bit of a spike right around sunrise, and then another peak kind of right around sunset. So we can kind of look at really kind of granular level data at a level that I don't think has really been possible in fish counting in hydropower facilities before. We're able to then kind of look at a total cumulative count over the course of a season. So what we're seeing here is we have our camera counts are in the tight bound within the middle there, and we've also got some of the other counts that have come from other partners counting in the same area, and they're extrapolated kind of error bound. So you can see ours are kind of much tighter compared to what we're seeing with our partners counting in there from a statistical sampling perspective. And then again, we can look at that kind of over the entirety of a season from a one dam or a one site perspective. So as opposed to looking at an entire cumulative, we can also see when do we see kind of spikes and when do we see a reduction as you see that migration start to kind of tail off there. As mentioned, we're going into our third spring now installing at Nova Scotia Power, specifically at White Rock, and some of the information that we've got from our partner at Nova Scotia Power in the value that they've seen from having this technology installed at White Rock is really getting that idea of the full season population dynamics of when those spikes are, when the fish are actually moving through the facility, and incorporating that along with an acoustic tagging study really provided a much fuller picture of the actual migration. So we were able to validate our newer technology doing these video counts with that acoustic telemetry to have some confidence in the data that's being collected from a relatively new product. And we were also able to look at some behavior and things that we wouldn't be able to capture with the camera. As mentioned, kind of such a small frame, a small area. The counting gives you an idea of number, but then really leveraging acoustic telemetry as a way to understand larger population movements and behavior around facilities. Some other things that we started to kind of really poke at and understand what are some of those insights that you can take from this data. Now that we're collecting this 24/7 data set at hydro sites, what can we glean from that information in terms of insight? So obviously looking at the fish per minute, the minute level granularity gets a really good view of day-night distribution, of peak travel times, of the quantity of fish they're trying to move at any given point. I think at one point during the migration at White Rock, we were counting upwards of 150 to 175 fish per minute moving through the ladder. Once we've had it installed now for a couple of years, able to leave it over winter, being out of the water like that, we're also able to get a particular date for a kind of first fish scene and the last fish scene as part of the season to really help inform some of those curtailment or shutdown discussions that Nova Scotia Power needs to have with their regulatory partners. We've been working with them to leverage that top of ladder, bottom of ladder count number to get an idea of what ladder efficiency looks like for fish trying to move through the system. So are we seeing a large number of fish entering the bottom of the ladder and then turning around and coming back out, and not seeing a kind of similar number of fish exiting at the top of the ladder? And then lastly, with the online dashboard, we're really able to keep season over season comparison, so we can see how are the counts for this season trending against the counts for the same period of time last year, and understand, are we seeing similar movements, are we seeing similar timing, or are we seeing a change to understand kind of why that may have happened. Rolling into 2025, so Nova Scotia Power has continued to expand the system within their network of hydropower here in Nova Scotia. So as of the end of 24, we had 10 dams with a total of 18 cameras installed across Nova Scotia. Starting in 2025, we have 4 more sites coming on board in Nova Scotia. We have a couple that have been installed in New Brunswick, and we're starting work with Ontario Power Generation in Ontario to start to develop a model for counting eels moving through a facility there. So some exciting developments coming in 2025. And lastly is really kind of just reiterating what we see are really kind of the value proposition of the Hydro AI product. So we think on the counting side, we can help save some of those labor costs related with trying to process the data that's being collected or providing fish counts currently, by providing that 24/7 fish counting and species identification. We can help improve regulatory compliance by providing all of that real-time data, those automated reports with the high level of accuracy that we've seen in our counts. We can help enhance the mitigation efforts to ensure that fish and the populations are able to move through and around dam infrastructure without impact. And ideally, hopefully, help increase green power generation by keeping hydro facilities online and being responsible and not impacting those fish habitats. And I think that's my last slide. It is. Email there if anybody has any questions that we don't get to today.
Thank you so much, Aaron. That was a great presentation.
Yeah.
Minh has his hand raised. Do you want to unmute and ask a question?
Yeah, Aaron. I really love the idea that you installed it straight inside the fish bridge there, so you can see it. Sorry, the fish ladder. But the question is, why do you not have a camera underwater? Like, what was the main restriction against having that? Because I think that having one underwater improves the water quality, not the image quality, much better than having it above, where the surfaces may may affect the water. Not to mention maybe sometimes even unable to see fish because they're maybe on top of each other. Yeah, so that's the question.
Absolutely, yeah. So I think we've absolutely learned that a camera on a side view, whether that's in the water or certainly have seen some fish ladders with kind of viewing windows on the side, is what we're going to need to do for species identification. I think with the herring ladders that we've seen on the East Coast, that top-down view actually is easier for just counting specifically, because then they can't be between, they're unlikely to swim on top of each other as opposed to kind of beside each other. So it makes it a little easier for that just overall general counting. Those particular ladders also don't get dewatered, which made it rather difficult to try and install a camera inside the ladder when it was kind of still full. We did try one year, we put one in, we couldn't get it really placed well, and we had a whole lot of bubbles and not a lot of great video coming from it, but absolutely agree completely. The next phase for us is understanding how do we install cameras with a side view for doing that better type of species identification work.
Can I clarify on that, Aaron? Is that they don't swim on top of each other because the water column is minimized there? Okay.
Absolutely, yeah. There's probably only about 12 to 18 inches of water kind of flowing over the top of that pool and weir style ladder. Yeah.
Okay, great. Thank you.
Right.
So we'll wait another couple minutes if folks have questions for Aaron. And then we will open up to questions for the whole group.
Yeah, Aaron, I actually have a quick question. You mentioned that Inova Sea is based out of Boston, Massachusetts. Are these same hydro AI systems being utilized in like New England as well?
Yeah, not. So we're just starting to kind of move outside of our backyard, I guess. We've had some great conversations with a group in Plymouth, Massachusetts. There's a dam there, and the town is doing some fish counting, similarly of river herring, kind of the alewife. So we have processed some of their video. They've got a really cool program. I don't know if anybody else is familiar with it. So same thing, they record video at their town dam, and then they actually post 1-minute clips of it on YouTube and crowdsource their counting. So if you've got some time to kill, you can go on their website and watch a kind of 1-minute video clip of a fish ladder, and you can tell them how many fish you counted as part of that. So we've taken their video for the last couple of years, have processed it for them so that they can then compare it to those crowdsourced counts. They're starting to build a new dam there in 2026, I believe. So we're going to be installing a system with them in Massachusetts. Yeah.
Cool. Very cool. Thanks.
Okay.
That's really neat.
Yeah.
Okay, folks, do we have any questions for any of the other presenters today, or of PNAMP or StreamNet staff involved in this? I'm thinking I should do a plug for the fish monitoring work group that PNAMP and StreamNet work on. We have a meeting tomorrow if you want to join us.
Great.
I'm not sure that Brice is still on. I suspect he has dropped off by now.
All right. I think we move on here, but these are great presentations. And like I mentioned at the beginning of this session, all of this has been recorded. So we will put it on the PNAMP YouTube channel. You can share it with your colleagues, and I'll send out an email to all those who participated or attended today, with the links to the YouTube and any other information. So look forward to that. I wanted to give a big thank you to our ETIS planning committee. There's a lot of us who are on this committee, and we've been working on this webinar series for more than a year now. So big thank you to everyone there. And then a little shout out to next week's session. So this was just session 2 of 5 today. Next week we have the data QA/QC session with 3 presentations. Again, it'll be a similar format. Each presenter will have around 30 minutes to present, and then we'll give them plenty of time for questions and answers. So look forward to that. It's the same time next week, 9 a.m. Pacific time on Wednesday. So yeah, look for that. And I think with that, we are done. But I will stick around for a couple minutes if anyone has questions for me or for the session or whatever.
Thank you, Sam. And a big thank you to our 4 presenters today. It was a really good session.
Thank you for having me. Great.